what this describes is the nature, another function of T helper cells, because in the 2 lectures ago I told you about these Th1, Th2, Th17 cells in inducing inflammation or for stimulating macrophage activation. I also said that these cytokines that these Th cells made were involved in stimulating B cells to undergo isotype switching. And this is what this lecture is about. How do T cells help B cells into making good quality antibodies or the right isotype of antibody for that particular situation? And with this comes this process called affinity maturation, which you need to know about. It's important because affinity maturation is the reason why vaccines have been so successful in medical science in protecting us from poliovirus, smallpox, etc. And these two processes, isotype switching and affinity maturation, are brought about by this important enzyme called activation-induced deaminase, or AID. And then, just to complete the whole picture of B cells and B cell differentiation, I'll talk about what happens once these antibody-producing B cells get help from T cells, how they develop into what are called plasma cells, which are fully committed antibody-producing cells, and then into memory cells. So, first of all, as I said, vaccines are very efficient, good vaccines anyway. And we know that when you introduce the protein antigen into a body, you can get a strong antibody response within 3 to 7 days after antigen exposure. Remember that I said that in primary, the first time you encounter the antigen, the antibody you will make will be IgM. And the naive B lymphocytes, which exit the bone marrow when they've undergone that clonal deletion to remove autoreactive cells, they then enter all the lymph nodes and the spleen, sampling for any antigens. If they see antigen, then they will make IgM from the outset. Now the thing is that when they see antigen in these lymph nodes, if they get help from T cells, they can then switch from making IgM to IgG, IgE, whatever, depending on the T helper cell that's been exposed to the same antigen in that site. But before we discuss that, we first need to, I think you've already done from an anatomy, the anatomy of the spleen and the lymph nodes, yeah?
Yes.
Right. But anyway, we'll revisit that so that, first of all, I told you in the pre-midterm, one of the pre-midterm lectures, about these B1 and B2 lymphocytes. So the B1 lymphocytes recognize polysaccharides, long-chain repeated molecules, and they don't need T cell help. And one of the first antibody-producing B cells in the developing fetus, they occur in the fetal liver, and they continue right up into adulthood. And they produce what are called T-independent antibody responses. So they don't need T cell help. They have broad specificity, but they're not as potent or as high affinity antibodies as the classical follicular B cell antibodies. Because they don't need antigenic stimulation to make these antibodies, these are called natural antibodies. So everybody will make these without having encountered antigen. Similar to the B1 cells in the liver, within the spleen and lymph nodes, in this region here, these are called marginal zone B cells, and they also make these broad specificity antibodies. So they are more like innate type B cell responses. They're quick to respond, but the antibodies they have, because it's IgM and pentameric, it's got high avidity, so it can just trap microbes once it encounters them. The strong high affinity antibodies are produced by these B cells, which are called follicular B cells. And these are the ones which need T cell help to make these high affinity, good quality antibodies. So these are the ones that we target when we are giving vaccination. We want to activate these follicular B cells. Now, so as I said, for B cells, you need T cell help. But now we have a problem in the body, because remember, when a T helper cell exits the thymus as a naive cell, it's specific for a particular antigen. When a B cell that has a surface IgM specificity for the same antigen exits the bone marrow, it goes through from one lymph node to another, but both the T and the B cell are in very small, 1 in a million lymphocytes have that specificity, right? So the chances of those two meeting up are very small. And so the question is, how do they manage to find each other? Do they go on dating websites or what, right? The other question is, when that appropriate T cell meets the B cell and starts providing help, how is that help only given to its corresponding B cell and not to other B cells which might have specificities for other antigens? And then thirdly, what is this help? What form does this help take? So before we talk about that, again, let's revisit the anatomy of these secondary lymphoid structures. So within the lymph node or the spleen, it divides into this outer cortical layer and an inner medullary region. Within the outer cortical region, you can find these things which look like half-cut boiled eggs, right? Some of them will have this inner dense core, others which will just look uniform. And these are the regions where B cells develop, your antibody-producing B cells, where the naive B cell, when it sees antigen, starts to proliferate and develop into so-called plasma cells. Within the medullary region is what's called the T cell zone, and this is where your naive T cells, which have exited the thymus and go from one lymph node to another, come here. And this region also is where dendritic cells, which have taken up antigen, also migrate, so that once they're there, they've already taken up that antigen, degraded it, and then processed it and presented it on class 2 MHC to those T cells. Alright?
So the dendritic cells inside the medulla?
You get dendritic cells inside the medulla, but you can also have, if this was peripheral tissue, dendritic cell might have picked up that bacterial cell and then migrated into the medullary region. Meanwhile, that bacterial cell, the same bacterial cell, would have moved into the bloodstream and come into this B cell zone where it's recognized by the B cells too. Right? So we've got the same bacterial cell, one picked up by the dendritic cells for presentation to the T cells. The other, the bacterial cell, is undegraded and just gets picked up by the surface antibodies of the B cells in this zone. So now, if you consider this B cell follicle, these areas of B cell growth here, and so we're talking about this border region between the B cell zone and the T cell zone, right? So this is the B cell follicle where all these naive B cells have collected. They've picked up this bacterial antigen and then bound onto the surface IgM molecule here. And remember, the B cells can be professional antigen presenting cells, so that protein antigen can be taken up by the B cell, degraded and presented on class 2 MHC. Meanwhile, the naive T helper cell in this region is seeing the same antigen, the same bacterial cell, which has been picked up and degraded by a dendritic cell, processed and presented on class 2 MHC, and stimulated that naive T cell into a fully functional effector helper T cell. So now you have both T helper cells and B cells recognizing the same antigen, but different epitopes, right? Because remember, your antibody molecule recognizes epitopes which are either exposed or internalized, but that is part of the native protein. The T helper cell is only recognizing these degraded peptides of that same protein that's presented on class 2 MHC. Right? So now these T helper cells, once they get activated by antigen recognition from the professional APC, they start to express a set of chemokine receptors which draws them to this marginal zone. At the same time, these B cells, having picked up this protein antigen, also express chemokine receptors which draws them to the same area. And this is what brings the specificity of a T and a B cell together through chemokine receptor interactions. Once they get together, now because the B cell has taken up that protein antigen, degraded this, and then presented the same peptide that T helper cell saw on the dendritic cell, these two can then get together and interact with each other through CD40 ligand interactions, which I talked about in the previous lecture, as well as CD28B7 cytokine receptor interactions. All right? So now with CD40 ligand interactions, both the T and the B cell are getting further stimulation of each other. And with this interaction, it causes that B cell to become this short-lived plasma cell where it churns out loads and loads of that antibody that it made originally from here. Okay? We talked about that. Now, once this interaction here happens, where through CD40 ligands, cytokines, and the presentation of that epitope on class 2 MHC by the B cell to the T cell happens, as I said, the B cell gets stimulated into this high-producing antibody plasma cell, but which is short-lived. But in addition, some of those T cells, which are effector cells, will differentiate even further into what are called follicular T helper cells. I talked about that in the previous subset thing. This is now where they appear. Before we go on any further, let's talk about this interaction here, where the B cell recognized this protein antigen via this epitope expressed on this protein and took it up and presented that same epitope to the T cell, which saw the same protein antigen but recognized a different epitope because it was presented by the dendritic cell, right? And this process is similar to what we understand about hapten carriers, because this epitope, which is recognized by the B cell initially, is similar to what you think of as a hapten. So in this example here, we have a B cell which is recognizing this yellow epitope on this protein antigen. Alright? Meanwhile, the T cell has seen this internal peptide folded within this same protein antigen, which the dendritic cell has degraded and exposed for presentation to the T cell. So both the T and the B cells are specific to the same antigen but different epitopes. Now once that T cell gets activated, and because that B cell, having taken up this whole antigen, can then present the same epitope on class 2, similar to what happened there with the dendritic cell, we call this linked recognition. So this is where you're going from the same antigen but different epitopes now to the same antigen and the same epitope. Okay? And because there's such close proximity to each other, this allows for direct one-on-one interaction in terms of CD40 ligand, cytokine stimulation, and so all of the other non-specific or the other B cells with different specificities aren't affected by this interaction. Only that epitope-specific B cell gets stimulated by that T helper cell. So this is how you get this direct specificity happening.
Doctor, does the body have T cells and B cells specific for a single antigen together in every single lymph node?
No, it happens, but not in every lymph node. So this is the hypothesis. Whether it's true or not, this is the best idea we've got. I mean, it seems to me the chances of this happening is quite rare. I mean, if you consider that one specificity of a B cell or a T cell is like 1 in a million. So basically, what are the chances of it's happening? But this is the best working model we have based on studies on mice. Right? Yeah, it's the same antigen. It's different epitopes. Yeah, but the same antigen. Yeah, because during the linked recognition, the B cell has taken up that whole protein and then degraded and presented the same epitope that the dendritic cell originally presented to that T cell. Alright? And did microbiology, have you learned about conjugate vaccines?
Conjugate protein.
So the Haemophilus influenza has a capsule, right? And the thing is that infants, babies, so in adults you have natural antibodies which can bind to the carbohydrate of Haemophilus influenza. But babies don't have many of these natural antibodies, so they can't protect themselves very well. Now the thing is that carbohydrate is a T-independent antigen. So to get a good vaccine, what they do is that they link the carbohydrate to a protein. And so that when they inject it in the body, the protein gets presented, the protein epitopes, which is the carrier, gets presented to the T helper cell. And that same protein carbohydrate complex gets taken up by the B cell. And so in this way you can artificially stimulate a T-independent antibody-producing B cell to that carbohydrate by stimulating T-B cell help. And this is the basis of what the H influenza conjugate vaccine is all about. And it's very successful. So we can manipulate this hapten carrier effect to create good T-independent antibody responses by making them T-dependent.
Does this allow development of memory B cells of those B1 cells?
So having stimulated T help, you can induce B memory cells.
With the T cells,
Yeah.
the T-dependent B cells that you just mentioned, could those also be memory?
Yeah. So now we complete the story. So with that initial TB interaction and that linked recognition, both the T and the B cell get stimulated to differentiate further. I said that those B cells get stimulated into the short-lived plasma, what they call plasmablasts, where they go into rapid proliferation, so large numbers, where they produce antibodies, but they only last for a few weeks. Meanwhile, some of these T cells which have been activated through the CD40 ligand will migrate into the B cell follicles. And so they will differentiate further from an effector T helper cell into this follicular T helper cell. And so now it's, again, this is through the new expression of chemokine receptors. So it draws these newly activated effector T cells into there to differentiate into these follicular T helper cells. In the meanwhile, some of these plasmablasts will also migrate into the B cell zones, into the follicles, and these are these dense cores, these parts, which are called germinal centers. So now within these germinal centers we have an interaction between these extra activated B cells and these extra differentiated follicular T helper cells. So now we're getting extra signals being bombarded between the two of them. Right? Another signal which is activating the differentiation from an effector T helper cell into a follicular T helper cell is this additional set of costimulatory interactions called ICOS, which is related to CD28, and it's called inducible costimulatory receptor. So you have ICOS ligand interaction in addition to the CD40 ligand. And within these germinal centers is where you have this process of isotype switching from IgM into IgG or IgE. Because if you recall in the immunogenetics lecture, I said that initially you get VVJ recombination to create your variable genes in your heavy and your light chain, and downstream of that you have your first C mu. So this is why IgM is always the first antibody receptor expressed. But with the aid of the follicular T helper cell, it has an enzyme, or induces an enzyme within the B cell called activation-induced deaminase, or AID. And what this enzyme does is it creates a loop between the rearranged variable region, so the antigen binding part of the antibody gene, and selecting out that particular isotype of antibody that it wants. So it nicks out that IgM, all those IgM genes. So now it's pulling in this example, the C region of IgE onto the variable region. So now you get IgE being expressed on this B cell.
Just to make sure, this happens in the DNA, not the RNA.
In the DNA.
So it's like, kind of like splicing, but in the DNA.
Yeah. And it's brought about by this activation-induced deaminase, which is stimulated by the follicular T helper cells. So remember, Th1 cells promote the expression of IgG. So a Th1 cell will undergo differentiate into a follicular Th1 cell to induce IgE production in the B cell. A Th2 cell will induce IgE production. And within the mucosa in the gut, you will get other Th2 type cells inducing IgA production of B cells within these follicles around the gut. So depending on the location and the type of cytokines will determine what kind of isotype antibody is made. Right? So all of that is due to this important enzyme called activation-induced deaminase, and this process of isotype is called switch recombination at the level of DNA. But the important thing to remember is that your antigen binding region, your VDJ, is not changed. All that's changed is the heavy chain. The constant region. But now, another important activity of this AID enzyme is that within the CDR regions of that VDJ antigen binding part, this same enzyme will introduce additional mutations within those epitope binding regions of that FAB fragment, the CDR regions. And by introducing additional mutations, it means that it changes the epitope binding specificity of those antibodies to increase the affinity of the antibodies for the epitopes. So now we've got a situation which is opposite to what I described in the removal of autoreactive B cells in the bone marrow, because now only those antibody surface expressing B cells which have the strongest stimulation for antigen, getting the highest signals, will outgrow the ones with lower affinity. So in this way, this enzyme AID is selecting out for B cells which produce the highest affinity antibodies. Right? So again, this is why vaccines are so successful, because by introducing a vaccine with either a carrier or an adjuvant, we are repeating this whole process of T cell help for B cells to create high quality, high affinity antibodies. And so once the B cells have undergone this somatic hypermutation, once they've undergone affinity maturation, they will develop into full-blown plasma cells, which are really long-lived, and they produce loads and loads of this high affinity antibody response. After the threat has been dealt with, some of these will differentiate into these long-lived memory cells, and these will then hang around within the lymph nodes to monitor. If they see that antigen again in the second time, they will respond very quickly and churn out these high-quality antibodies. So in summary, the type and the amount of antibody, first and foremost, depends on the type of antigen. So whether the dendritic cell picked up a helminthic worm or whether it picked up a bacterial cell determines whether you get a Th1 or a Th2 type response happening. In the same feature, the B cell is picking up that same antigen and processing that peptide so that with that close matching, it can present the same peptide to the T cell help to get that full set of stimulation to the B cell to develop into this high quality producing plasma cell. And so this is just a summary of the differentiation pathway of the B cell, going from a naive B cell expressing IgM. When it sees the T cell help within the extrafollicular zones, it develops into this short-lived plasma blast producing loads of these antibodies, but not necessarily high affinity. Then with the help of the follicular T helper cell, it develops into a plasma cell and into a memory cell.
Just one. Plasmablasts are dividing?
Yeah. So they're just expanding in numbers just to make lots and lots of antibody. This just gives some extra information, like these germinal centers develop about 4 to 7 days after that initial interaction between the T helper cell and the B cell. And these are the set of signals or receptors that the follicular T helper cell expresses, namely this important co-stimulating molecule ICOS, which engages ICOS ligand on the B cell. And so this is what drives affinity maturation and isotype switching. IL-21 is an important cytokine that follicular T helper cells make, which is needed for the germinal center development and differentiation of B cells into plasma cells. As I said, whether the B cell antibody that's made is IgG, IgE, or whatever, is dependent on the cytokine produced by the follicular T helper cell. Alright, that's it.